Thank you so much, and thank you for giving me the opportunity to be here, at least virtually. I'm really looking forward to having a discussion with you all about some issues, specifically around the similarities and differences between the way biology and technology processes information. And I'm going to go fairly quickly over some of the biology. If you want to dig into the details, everything is here, and then these are kind of my own personal thoughts about what I think some of this means. So we all know who Alan Turing is and how he was interested in the fundamentals of intelligence in diverse embodiments. And he thought a lot about how to implement different kinds of minds in novel architectures. And in particular, he was interested in reprogrammability and thus intelligence as a kind of plasticity. What's not as frequently appreciated is that he also wrote this amazing paper on the chemical basis of morphogenesis, and this goes into mathematical models of how embryos self-organize from initially a randomly distributed set of chemicals. And so now, why would somebody who is interested in computation, in programmability, and intelligence, why would they be interested in the early stages of development? I think it's because Turing saw a very profound truth. Which is that there's really a deep symmetry between the self-assembly of the body and the self-assembly of minds. And we have studied now for a couple of decades the various parallels between those two processes, and I think they're fundamentally the same process. Because what happens in biology is that life makes a journey. Each of us took this trip from being a unfertilized oocyte. So this is a little bag of quiescent chemicals that people would look at and say, well, this is well handled by the laws of physics and chemistry. There's no need for any kind of concepts from intelligence or cognition or anything else. But slowly and gradually, this becomes something like this, or even something like this, a system that will certainly make claims about being cognitive and having beliefs and memories and goals, and perhaps even not being a machine and things like this. So what's important here is that the hardware, the biological hardware, is able to autonomously make this slow and gradual transition. And by the way, this is not the end of the story. There are some many interesting things, both pathological and just really strange things that I'll talk about in a little bit that can happen even after this journey is complete. And so one thing that I want to point out that's important for deriving insight for AI and things like that from this sort of process is that the standard story of biology that you may have heard is incomplete in important ways, and it's being revised by a number of people, including our lab. But what's important to know is that DNA is not a program for what to make. Okay, DNA is not really a description of the final product that we're going to get here from any of these kinds of cases. And what we study in our lab is a model system, which is groups of cells navigating the space of anatomical possibilities, so all of these different possibilities, as a model system for collective intelligence. And I think this is very important, because what it reminds us of is that we need better theories of transformation and scaling, not ancient magical categories such as humans and machines or living things versus computers. I think those really hold back science in important ways, because all of us started life as a single cell and chemical networks before that, and eventually, both on an evolutionary scale and on a developmental scale, slowly, gradually, without any special jumps or discontinuities, we become this kind of modern human that features in all of the discussions of philosophy of mind and things like that. But not only is this a continuum, but also now we know that both biologically and technologically, there's a very wide range of different kinds of chimeras possible, and many of these already exist. And so the reality is that both we and the kind of technologies that we make are, at the bottom, made of some of the same stuff. Some of the details differ here, but fundamentally we are all composed of matter, and the trick is to understand how different kinds of observers interact with different kinds of systems to evince agency and the kinds of things that we're interested in terms of intelligence. This is as much a problem for biology as it is for robotics, for computer science, for artificial intelligence, and so on. So what I would like to do is to develop a framework that allows us to recognize, perhaps create, but most importantly, ethically relate to truly diverse intelligences, regardless of what they're made of or how they got here. And so this means, of course, familiar creatures such as us and birds and maybe a whale or an octopus, but also some really, really weird organisms, colonial things, swarms, synthetic biology, so engineered new life forms. I'm going to show you 2 of those today. Of course, AIs, whether software or robotic, and perhaps even someday aliens. This, broadly speaking, this is a program in the field called diverse intelligence, which I think is very important and really underappreciated as the implications that this field has for AI and AGI and things like that. Of course, I'm not the first person to try for a framework like this. Here's Wiener and his colleagues back in 1943 trying for some kind of a scale from passive matter all the way up to human and beyond human intelligence. But what we're interested in using various model systems, including cells, navigating anatomical decisions, to understand how exactly the scaling works. Okay, how do you get from simple matter to these kinds of cognitive things? And in order for me to claim that this is that development and regeneration and those kinds of things are not simply the mechanical feed-forward emergence of complexity from simple local rules, I would have to show you that what we're actually dealing with is context-sensitive plasticity. In other words, intelligence, the way William James defined it, being able to reach the same goal by different means. Okay, so we need to talk about what exactly does it mean to navigate this morphospace in a way that is something other than simply feed forward some sort of cellular automata-like structure. And you can see all of this described in careful detail here. I'm going to go through fairly quickly to give you an overview of some of the biology that underlies some of these ideas. What you're looking at here is the tadpole of the frog, Xenopus laevis. Here are the nostrils, the mouth, the spinal cord is here, the tail, the gut. This yellow thing is the gut, and then this whole thing is the head. Now, what you'll notice is that we've prevented the 2 primary eyes from forming, but there is an eye that we've caused to form on its tail. And that eye makes an optic nerve. The optic nerve comes out. It starts to look. Sometimes its synapses on the spinal cord, sometimes on the gut, sometimes nowhere at all. And it certainly doesn't hit the brain. It never reaches the brain. And yet, as we discovered by building this machine that automates the training and testing in behavioral light responsive training assays, these animals can see. And so what you find is this amazing ability for the radical refactoring of the sensory motor architecture. For the first time in millions of years, the eyes are not in the head, they're not connected to the brain, they're somewhere else. And yet this animal does perfectly well right out of the box. No new mutation, selection, evolution, adaptation. No need for any of that. Out of the box, you change the configuration, it works perfectly well. They can see. It gets even more impressive as caterpillars, which you can think of as a kind of soft-bodied robot with an appropriate controller for running a soft body that moves in 2-dimensional space and eats leaves. It has to become this butterfly, this hard-bodied creature. You need a completely different controller for that. It flies now, and in order to get from here to here, what happens during metamorphosis is that the brain is basically dissolved. So most of the cells die, most of the connections are broken, and then it builds an entirely new brain suitable for this kind of lifestyle. What's amazing is that if you actually train the caterpillar, the butterfly or moth will still have the information, but retention of information past this drastic refactoring is not the amazing thing here. It's amazing enough that it would be nice if we had architectures where you could scramble the hardware and the memories would stay intact. But it's actually more amazing because the specific memories of the caterpillar are of no use to the butterfly. If you train it to eat leaves on a particular kind of stimulus, to crawl and eat leaves, the butterfly moves differently. It doesn't crawl. It doesn't care about leaves. It wants nectar. So there has to be not only a generalization, but a remapping of the specific experiences that this creature had onto an entirely new body. So what's actually happening for these memories to survive is they have to be remapped onto a new architecture. And this requirement to maintain not the fidelity of information, but the saliency of information for novel scenarios is, I think, what underlies the magic of biology and the things that we can learn from it in computer science. There are many kinds of plasticity. For example, if you take this tadpole and you scramble the craniofacial organs, right? So this eye is on top of the head, the mouth is off to the side. It's like we call them Picasso tadpoles. Everything is in the wrong place. they will still make quite normal frogs because all of these organs will move around in novel paths. They'll get to where they're going, and then they stop. So you see this, that genetics is not specifying a set of hardwired steps for these. What it does specify is a system that can do error minimization. And it can do this kind of, means ends analysis kind of thing where it will keep moving until it gets to a particular pattern, and then everything is satisfied. Not only that, but these kinds of systems they're reliable for sure. So this acorn all over the world, you see these things reliably in billions and billions of cases just building the same exact structure. So it sort of lulls us into a false sense of understanding that we say, well, we know what the oak genome knows how to do. It knows how to build this particular structure. But along comes this non-human bioengineer, which is this little wasp. And what the wasp does is prompt the cells with new information that hijacks their morphogenetic ability and causes them to build these galls, these amazing structures. We would have had absolutely no idea that these cells are able to, besides the flat green thing that they always make, could even make such a structure. We would have no clue if it wasn't for this creature that figured out how to get the desired behavior. And these are not made of wasp cells; they're made of plant cells. So this is kind of a minimal prompts that hijack the ability of this thing to build many things beyond what it normally reliably builds. So what I've shown you so far is this plasticity and of novel outcomes, and of course such plasticity is hackable or reprogrammable. Now, we are all a kind of collective intelligence because we are all made from agential materials. This is a single cell. Okay, now this is a free-living organism called the Lacrimaria, but you get the idea. This single cell, it has no brain, it has no nervous system, but it is incredibly competent in all of its tiny little goals. It has a little cognitive light cone, and it's very competent in those goals. We are made of parts with agendas, okay? And this is quite different than most of the technology that we build. You don't need to worry about robot cancer because they are generally speaking made at a single level where the parts don't have their own agendas. They're not going to decide to defect and go off and do something else. The collective does not need to worry about constantly binding its parts, hacking and controlling its parts towards common purpose. This is what biology is doing all the time, 24/7. Because we are made of parts that otherwise would do their own thing. And in fact, even below the level of that single cell, the material of which the cell is made, so the chemical networks inside, already have 6 different kinds of learning that they can do. Okay, just by virtue of the mathematics of the way that these networks work out, it turns out that they can do habituation, sensitization, and associative conditioning. Okay, so the material itself, long before you get to cells or brains or anything like that, the material, the chemical material that we're made of already has the ability to form memories. And by the way, it turns out that as they're trained, as they form these memories, causal emergence goes up. Okay, so the system is actually reified as a high-level system by virtue of the things it learns, right? And we are currently in our lab trying to take advantage of this kind of thing for drug conditioning and various medical applications. So what's happening in biology then is that unlike most of our architecture, it is a multi-scale system like this, not just structurally, but the fact that every level has the competency to solve certain problems in various spaces, in their own spaces. And that means that each level has to bend the option space for its parts and keep them aligned towards goals that the lower level knows nothing about. And so here we come to an interesting point about embodiment. We as humans are, because of our own evolutionary history, we are very obsessed with three-dimensional space, and we think that three-dimensional space is the real one, and then, mathematicians are good at thinking about some other spaces, but, 3D space we think is the real one. And we are pretty good, not great, but we're pretty good at recognizing intelligence of medium-sized objects moving at medium speeds in 3D space. But biology has been solving problems by navigating spaces long before muscle and nerve evolved. And so we have to really understand that this perception action loop that's the basis of all agency happens in many other spaces. And so biology does it in the space of gene expression, in the space of physiological states, and what we're talking about now, anatomical morphospace. So I think this is important because a lot of people we'll say that if it's not driving some sort of robot that wheels around your your kitchen or the outside environment, that it's not embodied. And I think that's a big mistake. I think both in robotics and in biology, a lot of the organoids that look like they're just sitting there, and a lot of the software AI agents that look like they're just sitting there and not engaging with the real world, are actually very likely navigating many other spaces that we're just bad at noticing. And within this anatomical morphospace, we see a number of things. We see things like regeneration, where, for example, this axolotl, this amphibian, you can cut anywhere along this line, and the cells will immediately grow what they need to do, and then they stop. And that's the most amazing thing about this, because they can sense they've been deviated from the correct place in anatomical space. They will work very hard to get there. When they get there, they know they've gotten there. They can sense that the error has fallen below acceptable thresholds, and then they stop. Right, that stopping point is really critical. And it's not that any particular cell knows what a finger is or how many fingers you're supposed to have, but the collective absolutely does. And you know that it does because you know that it will stop when it gets to the right point. And as I'll show you in a minute, that set point is actually rewritable. It's reprogrammable. Now, it's not just about controlling or repairing damage. It's really about the top-down control of high-level goals onto the hardware. So what you're seeing here is another experiment done back in the 50s, where people would take the tail of an amphibian and surgically attach it to the flank. And what you see here is that over time, it slowly transforms into a limb. Not only does this thing transform into a structure more appropriate to its new location, but the cells at the tip here, I mean, in their local environment, they're perfectly correct. They're tail-tip cells sitting at the end of a tail. But what happens is that they start to turn into fingers. And that's because this large-scale pattern, okay, the error that is sensed by the system when you do this grafting experiment, propagates all the way down to control not only the cell behavior, but the molecular biology, the new genes that need to be turned on and off to make the whole thing more coherent, right? So talk about alignment, right? You have to get the cells and you have to get the molecular pathways aligned towards this large-scale body plan of which actually they know absolutely nothing. You have to have an architecture, which biology does, that propagates these signals all the way down and make sure that the low-level details are feeding this high-level anatomical journey that the whole system wants to take. Now, I don't have time to show you all this, but one of the things that is kind of a central new thing that our lab has done is to find out that very much like in the brain, the exact same mechanisms, which we call a kind of cognitive glue because it binds subunits towards large-scale goals, the same mechanisms are used in the body as are used among the neurons of the brain to allow you to navigate space towards specific goals. In other words, if you ask, how is it that we have this amazing neural architecture that allows us to pursue goals in the future, have memories of the past, and so on, it turns out that brains got that trick from very ancient bioelectrical signaling that was here since the time of bacterial biofilms. And this is just one example. We've learned to visualize in non-neural cells all the electrical activity that's happening here. And I'm going to show you a couple of cool examples. So what's happening here is these are two frames. These are two frames of a video, and the colors here represent cell voltage. Okay, so these are slowly changing voltage states, just like they study in electrophysiology, but slower. And the colors are different voltage. So there's this cell, and what you'll notice is that it starts off with this voltage represented by the red, and as soon as it touches just a little bit, it touches this whole mass. Bang! It gets hacked. And so I'm going to show you this is the video. So here it's got its own voltage. It's moving along. Now this guy's going to reach out. Boom! That's it. It's been now convinced that it's going to become part of this group, and it merges, and that's it. These bioelectrical states are controllers, not just indicators, but controllers of whether cells are going to go off to do their own thing or join a collective and work on these very large-scale goals. And we can now track these messages that are able to hack them. Not only can we track them, but we can reproduce them and bend them to our own purposes. So for example, when I change the bioelectric state of specific cells, and this is not by electrodes, or there's no frequencies, no magnets, nothing like that, we basically hack the native interface that the cells are using to control each other, which means ion channels. So you change the ion channel properties in these cells, and we can put in a little pattern that says make an eye here. And sure enough, there's an eye in a place where the gut was supposed to be. These eyes have all the right internal structure, lens, retina, optic nerve, all this stuff. And so now this is really important, talking about this multi-scale architecture, we did not have to control any of this. In fact, we have no idea how to micromanage the cells and tell them to make an eye. Eyes are very complex. We don't know how to do that. What we do know how to do, much like you would with any cognitive system, is to give a high-level prompt that the rest of the material interprets in ways that propagate down to the parts to enable them to do the thing that we're supposed to do. We're doing it right now. For me to get you to remember what I'm saying, I'm not trying to reach into your brain and change your synaptic proteins. You will hopefully do that on your own. We are communicating through a very narrow interface with very sparse kinds of messages, and so we can do those kinds of things. And the cells that we inject, so here are these blue cells, will actually go on and recruit all these other cells who we never touch directly to help them build this larger scale structure, right? So we instruct these cells and we say build an eye, and that's really all we say is build an eye, and that they instruct everybody else and get them to do the right thing. We can do that. We can do many other things. For example, here are flatworms, so they normally have a tail and a head. And they have a bioelectric pattern memory, not in the brain, in the entire body, that tells them how many heads a proper worm is supposed to have. And so we can now visualize that. I could show you all these things, but there's not that much time. But just to say that we can visualize that set point, okay, the pattern to which they repair, and we can rewrite it. And we can rewrite it in a way that says, no, you should have 2 heads. And if we do that, then when we cut off the head and the tail, boom, you get these 2-headed animals. This is not Photoshop. It's not AI. These are real animals. You can see them here. And the amazing thing is that when you then cut these things, they will continue to regrow as 2-headed. It's permanent because, like any good memory, it holds. Once you've changed the set point, it remembers and it holds. And so what's critical to understand, the reason I'm showing you this is because there's been no genetic change here. There's nothing wrong with the hardware. You can sequence these guys all day long. None of the tools of modern molecular genetics will reveal that, in fact, these guys are going to regenerate as 2-headed, because that's not where the information is. And so you can see that the material is it uses these physiological circuits to store information that does something different than the default outcome that this hardware normally does when it comes into the world. And so I've now shown you many examples of this amazing reprogrammability and plasticity. And so it's time to take some guesses as to why this occurs and what it is that's special about the biological architecture that maybe we can learn from in making AIs. So what I think is happening here is that both on a cognitive and a morphological level, the following phenomenon is taking place. Just consider that you don't have access to the past. What you have access to at any given moment is the engrams, the memory traces that have been left in your brain or body by past experience. So we are a collection of these kinds of selflets, and many of them are not changed that much. I mean, okay, for the butterfly caterpillar thing is pretty drastic. In puberty, mammals have a pretty drastic as well change, as certainly humans do. But fundamentally, most of these things are similar. But nevertheless, what's happening here is that at any given moment, you don't know what your memories really mean. In fact, it doesn't matter what they meant to past you. What matters is how to interpret the information that you got for the most adaptive story going forward, the thing that will tell you what to do next. And so I'm sure you'll all recognize this kind of architecture, and it's described in detail here, where what's happening first is that your experiences and the results of them are getting squeezed down, right? They're getting compressed and generalized into this thin, very, very sparse representation, and this is the memory medium inside of cells. And then continuously it has to be decoded. In other words, what does this set of molecules or this bioelectrical map, what does it mean? Can we decode it and find out what we're supposed to do? And so this part, I think, is algorithmic and it's deductive, but this part I think is fundamentally creative because you've lost information here. Okay, you don't know what it meant to your past self, and it actually doesn't matter. And so the result of this is that life isn't overtraining on its evolutionary priors. Whether it's memories in your mind or genetic information in your cell, all of it is basically just a message from your past self. And as all messages, it is now up to you to interpret it in whatever way you want. And that is not only a kind of freedom, but it's actually a kind of really important responsibility, because you have to do this. You have to do this continuously. And so that gives rise to this idea of morphogenesis as a kind of beginner's mind where you can't really assume very much. I'll just give you a simple example. This is a cross section through a kidney tubule. Okay, so you see 8 to 10 in a newt. So you see 8 to 10 cells usually work together to make this. One thing we can do experimentally, and this isn't my work, this is a very old work by Fankhauser. You can make cells gigantic. You can force the cells to be very large. And if you do that, what you find is that it adjusts. If you make them large, it will make fewer cells will make the same thing. If you make them huge, one cell will bend around itself and still give you the same structure. This is the classic thing that they test on every IQ test. We give you a set of objects, and you try to solve a problem you've never seen before by creative use of the things at your disposal. The way you do this is actually you give it way more copies of its chromosome number, and then they do this. So it's able to get the job done. It's able to take the journey, even when it's dealing with completely different parts, because it's able to take new affordances from the molecular hardware that it has and use them appropriately in a context-sensitive manner to solve a problem it has not seen before. And so think about what this means when you're a newt coming into the world. You don't know how many copies of your genome you're going to have. You don't know how big your cells are going to be. You don't know, never mind the environment, you can't even trust your own parts. And this is the thing about biology, is that, and actually Josh Bongard had some fascinating work on robots that also don't know exactly what their structure is. They have to figure it out from scratch, and this is now quite old original work. So the thing with biology is this. We are in at least in most computational architectures that I'm aware of, there's a lot of redundancy. There's a lot of error correcting codes. There are abstraction layers to make sure that you are keeping the fidelity of information. When you program in high level languages, you don't worry about your silicon and your copper going off, or your bits out of the registers are going to kind of float off. You don't worry about it. But biology is completely different. It assumes the hardware is unreliable, because it is unreliable. It's going to be mutated. The environment often changes. You don't know how many copies of any protein you're going to have. Things degrade. And so it's forced to go through this bottleneck where it doesn't just, and the same kind of architecture is here. We don't make other organisms like us. We make eggs. And those eggs then it's on this material to figure out what this information is and what to do with it. And so what's happening here is that I think there's this amazing ratchet, this amazing intelligence ratchet between the fact that competency and the ability to improvise what your memories mean for the future actually hides a lot of information from selection. Right, and as you do this, evolution has no choice, but it has a hard time figuring out the good genomes from the bad, and so a lot of the work is done on actually cranking up this competency, and so you get this positive feedback loop because of the fundamentally unreliable material. But just to finish this off and hand it off to Hananel in a minute, one of the most interesting things is that it's not all about evolution. In other words. Here, I'll show you a couple of synthetic life forms. So first of all, these are xenobots. These are made from frog epithelial cells. And not only do they self-assemble into these little motile kinds of forms, but if you give them a bunch of loose skin cells, they will also run around, collect them into little piles, and those piles mature to be the next generation of xenobot. And guess what they do? They do exactly the same thing. So we call this kinematic self-replication. It's kind of von Neumann's dream of a machine that goes out and builds copies of itself from things it finds in the environment. But as far as we know, no other creature on Earth replicates by kinematic self-replication. This is completely novel. Not only that, if I show you something like this and I say, what is this, right? What do you think this is? And people will often say this is it's some sort of a primitive organism that we got from the bottom of a pond somewhere, and it should have a genome like that. If you were to sequence it, you would find the genome of 100% Homo sapiens. These are adult human tracheal epithelial cells that self-assemble into this motile little creature that looks like no stage of human development. It has its own lifestyle. It has if you look at the gene expression, it has more than 9,000 differential transcripts, so its transcriptome is completely different than those same cells. We've done nothing to the DNA. There are no synthetic biology circuits, there are no scaffolds, there are no nanomaterials, there's no genomic editing. All we've done is allow them to reboot their multicellularity, and you get you get proto-organisms. You can see them here that self-heal. They have a completely different set of gene expressions. They're actually younger than the cells they came from, so they also roll their age back, and they have this remarkable property. If you let them swim down a big scratch that you've made in a bunch of human neurons, you will find out that they actually assemble into the superbot and they heal. They heal across the scratch. Here's what it looks like when you lift them up. So they have these amazing properties. But you know, who would have known that your tracheal cells that sit there for decades quietly dealing with mucus, that they have the ability to do this? And so we have a fundamental question, and then I'll wrap up here, which is, we know when the computational cost was paid to develop frogs and frog embryos, right? So millions of years of that genome bashing against the environment computed the correct way to survive. When was the cost paid to develop xenobots or anthrobots? There's never been any xenobots or anthrobots. There's never been any selection to be a good xenobot or anthrobot. So I'm claiming here that not only the standard version of biology is useful for us in AI research, but actually there's something extra that's happening here, which is that it's not just physics and it's not just genetics. We already know, I mean, biologists love these two things, right? Everything in biology needs to be explained by a history and by some physics of the environment. That's it. That's supposed to be the exhaustive list. But we know that there are mathematical objects, such as here. This is a Halley plot made by this very simple formula in complex numbers. Look at the incredible structure. None of this is set by any part of physics. None of it is set by any kind of a history of selection. It comes from the same place that the laws of mathematics come from. And I think it's important that physics doesn't affect, but it is affected by, specifically constrained by, mathematical patterns, right? There are many aspects of physics that if you ask why is this the way it is, it's because this mathematical thing has certain symmetries or whatnot. But biology also doesn't determine the laws of mathematics, but it exploits them. It exploits a ton of free lunches that are provided by these things from this other space. And so my claim is that we should be exploring this space, much like mathematicians do, and that biology offers novel exploration vehicles, so xenobots, anthrobots, all this other stuff, that allow us to explore this incredible latent space of form and function that provide a lot of free lunches that are not explained by either the materials, the history, the algorithms, and all of that. And I think there's a real parallel here between the relationship between minds and bodies and the relationship between mathematical objects and the physics that they inform. And I need to stop after one last slide, but I'm just gonna point out that if you want to take a look at this slide, a lot of people think that, okay, fine, there are these amazing and people will call them emergent. I don't like it, but a lot of these amazing features recognizable by behavioral scientists that emerge in complex structures. I don't think complexity has very much to do with it. You don't need to be alive. You don't need to have huge complexity. Even bubble sort. Okay, so if you look at this, even things like bubble sort have unexpected side quests that they do that are readily recognizable by behavioral scientists that are not in the algorithm. It's easy. The algorithm is 6 lines of code. Everybody can see what it is. They're doing things that are not in the algorithm. And so I think this is kind of tongue in cheek, but the idea is, there are patterns that are informing what happens in the physical and biological world, but they are not uniquely for us or even for living things. I think they kind of go all the way down in terms of complexity. So this means that because of the plasticity and the kind of interoperability of life, all of current living things that Darwin was impressed by when he said this are here. They're a tiny corner of this incredible option space, and pretty much any combination of evolved material, engineered material, software is going to be affected by the ingression of these patterns. And they're not just about distributions of primes and things like that. I think some of these patterns are basically behavioral propensities or kinds of minds. And so we are going to be living in a world with hybrids and cyborgs and chimeras and very unconventional creatures, and we need to radically expand the way that we understand embodiment in other spaces and behaviors that have intrinsic intrinsic properties, not things that determined by what we think we've built. And so the summary here, I'll just point out that I think the road to AGI is not about brains. It's not about neuromorphic computing or three-dimensional embodiment. There are some very deep concepts that we've talked about, including this improvisation and so on. Just as biology's story of the genotype to phenotype map doesn't capture what's actually going on in the problem-solving intelligence of that process, I think computer science's pictures of algorithms also does not tell the whole story. I think this notion of, we made it. I know what the algorithm is. I know what all the pieces are, and so I know what it does. I think that's profoundly wrong in a much deeper way than just that it has unpredictability or unexpected complexity. And I think, much like Magritte was reminding us that our formal models of things or our pictures of things are not the things themselves. Here's the computer science version of this. This is not a Turing machine, because I think nothing, not even simple algorithmically driven machines, are actually fully encompassed by our formal models of them. And so I think in important ways, biological bodies are basically a kind of thin client for certain patterns that come from this Platonic or latent space. I don't think we make intelligence. I think what we make are embodiments that facilitate the ingression of these patterns. And these things may or may not be anything like the natural minds that we have. The stuff that we force them to do, such as talking and language, may have potentially nothing to do with some of their intrinsic motivation, and we need to understand this. So this is the research agenda, is to try to understand that mapping between the physical pointer that we make and some of the patterns that come through. So I'll stop here and just thank the people who did the work that I showed you today, and we have lots of amazing collaborators. And so the next part of the story is going to be told by Hananel Hazan, who's actually got some interesting computer science that goes in this direction. So